This lecture will go over some of the main research techniques that we use in neuroscience. My background is neuroscience, so some of these techniques I have personal experience with. If you have any questions, feel free to ask. So how exactly can we study the brain? The first place to start is to think about what you know about the brain and what kind of device might be useful for figuring out how the brain works. What would you want this device to measure? Well, if you think back to the book and your readings and to the earlier lecture in this module, remember that the brain has different kinds of signals. There's an electrical signal and a chemical signal. We need to figure out how to measure those. One of the things you probably learned from your book is that the back of the brain processes vision. How do we know that? So we're going to go through 4 main techniques that are used in neuroscience to figure out what our brain does. The first technique is called electroencephalography, and this technique is also known as EEG. And EEG measures electrical signals from groups of neurons. So if you look at the picture on the slide, what you see is a person that has a whole bunch of little recorders all over their head. These recording devices are called electrodes, and he has probably somewhere between 15 and 30 of them on his head. A lot of EEGs can have up to 100 to 200 different electrodes all over the scalp. EEG is very easy. It's what's called non-invasive. That is, they don't have to drill into you or anything like that. The little electrodes just kind of get taped onto your scalp. And these electrodes are going to measure any electrical activity that's occurring. So if it's occurring at the front of the brain, in the frontal lobe, the recorders that are towards the front of his head will be active. If it's occurring towards the back of the brain, in the occipital lobe, then those recorders are going to be active. This is usually the kind of data that you get from an EEG. EEG is used very frequently in sleep studies. So what you're looking at here is the different stages of sleep and the brain waves or the electrical waves that are going on in each stage. Notice that the excited brain on the top has very short, fast brain waves. The relaxed brain gives you longer, slower brain waves. Notice that as you go from drowsy to deep sleep, you get longer and longer waves. Don't ask me about coma. We don't really understand what's going on there. Any thoughts on which one of these is the closest to what a brain is like when it's dreaming? I'll give you a second to process that. And the right answer for that is actually the excited brain. When you have a dream, your brain is actually very, very, very active. 2 of the other techniques that we use are shown here. The first one on the left is called a PET scan, and the one on the right is called an fMRI scan. You may have had an MRI on your body before if you've ever broken an arm or a leg, but this is an fMRI. It's not just a regular MRI, which will show you bones and so on, but the F stands for function, so it can actually show you the activity of certain parts of your body, and specifically your brain. How do these work? Well, they look at blood flow. If you think about using a muscle, if you were to work out and flex your bicep a lot, the blood goes to wherever in your body is active. So we believe that blood flow equals activity. So if you're looking at something, then the area in your brain that's most active would be the visual areas. And so if we track where the blood goes, it should go to those visual areas. Here we've got a picture of a brain. Notice you've got the picture on the right, looking from the side on the right. And we've got the one on the left, which is the view from the left. So let's see if we can figure out what this person was doing. The colored areas in the brain show where the activity was. Notice on the left side there are activity in the temporal lobe that the arrows are pointing to, and also in the frontal lobe that the arrows are not pointing to. In the right side, there's also temporal lobe activity. So what's happening to lead to all this activity? Well, the temporal stuff, as you probably can figure out, deals with hearing. So this person was hearing something, and they were hearing it in both ears, because notice that both temporal lobes are active. Then this person was asked to respond to whatever they heard, so they had to push a button. Can you figure out which hand they had to push the button with? So remember, movement is processed in the frontal lobe, and because their left frontal lobe is active, that means they were moving their right hand. For the most part, if you're running a study with an fMRI or a PET scan, you know what your subject was doing, so you don't have to play detective and figure it out. But it's nice to be able to look at a brain and try and solve a problem, which is largely what we're trying to do. The last technique that we're going to talk about is a fairly new technique. EEG, fMRI, PET have all been around for 20 to 50 years. TMS has been around for 10 to 15 years. TMS stands for transcranial magnetic stimulation. And for this technique, they actually use a magnetic wand to target a certain brain area and send pulses through your skull. As shown in the picture, the wand on the guy's head is able to essentially zap the guy's brain. If you dissect the word, remember this is called transcranial magnetic stimulation. Transcranial means across the head. Magnetic stimulation tells you what it's doing. So if you know the name of this technique, it essentially tells you what it does. So we are essentially stimulating the brain with magnets. With this technique, we can do two things. We can either send short, strong pulses and excite a certain area. So for example, if I zap your movement area for your hand, you would move your hand. Regardless of whether or not you meant to, your brain would simply be activated and you would do it. If I zapped your visual area, you might actually see something, probably a little blob, something like that. If I zap your auditory area, you may hear something. It probably wouldn't be anything intelligible like a word or something like that, but maybe more like a tone or a funky type of static sound. If I zapped your touch area, you would feel something. So that's one thing that this technique can do. The other thing that you can do with TMS is send a weaker, longer pulse and inhibit an area. You can imagine that if I did this on your movement area, you would try to move, but you couldn't move. It would almost be like having a temporary hole in your brain. We do think it is very temporary. Usually this seems to correct itself within about 10 to 15 minutes. That's the end of the research techniques on the brain lecture. Again, please let me know if you have any questions. Since this is my background, I have a lot more stories and explanations that I could give if you need help understanding them.